Transcription. DNA is composed of molecules called nucleotides. There are 4 different nucleotides: adenine, thymine, guanine, and cytosine. The order in which these nucleotides are arranged determines the genetic code. Since DNA is two-stranded, when they are together on the ladder, the nucleotides point inward and form base pairs. Adenine pairs with thymine, and cytosine pairs with guanine. When DNA is copied into RNA, the chemical base that pairs with adenine is uracil instead of thymine. The ladder of DNA, when going through the process of transcription, must unwind so that RNA can be made. One of the 2 DNA strands is used to be replicated and create an RNA, or coding strand, similar to the DNA strand, after the DNA has been unzipped by the enzyme helicase. These 2 strands are anti-parallel, meaning the 2 strands run in opposite directions, with the 3 and 5 ends of one strand running opposite to the 3 and 5 ends of the other strand. To initiate transcription, a transcription factor, a protein that initiates and controls the rate of transcription, binds to the promoter region of the DNA, near the start of the transcription site. The RNA polymerase, an enzyme to help transcribe DNA to RNA, builds the RNA strand using the 5 to 3 direction and can only add nucleotides to the 3 side, starting from the promoter. The RNA polymerase will keep copying until it reaches a sequence of DNA called the terminator. The product is a strand of pre-mRNA. Before pre-mRNA can turn into messenger mRNA, it must go through a series of steps. First, a methyl cap and a poly A tail are added to the front and back of the pre-mRNA respectively. These protect the mRNA and help it get from the nucleus to the ribosome so the transcript is not broken down. Next, introns, segments of RNA that do not code for proteins, are removed in nRNA splicing, leaving only the exons, the important parts of RNA molecules necessary to code for the protein. As the transcription process is coming to an end, the translation process gets to work. Translation. Translation is when the RNA strand is decoded to build a protein, which has the same specific amino acid as the DNA. A finished RNA strand is called the messenger RNA or mRNA, and is what gets translated. Transfer RNAs, or tRNAs, are used to connect the RNA codons to specific amino acid sequences using anticodons, which are found on their ends. mRNA strands get attached to ribosomes, which start translating the amino acid sequences in groups of 3s called codons. The ribosome then reads the mRNA and tRNA with the opposing anticodon. Then a set of 3 nucleotides that binds to a matching mRNA codon through base pairing arrives at the ribosome, bringing the correct amino acid for that codon. The ribosome keeps shifting over a codon to be read until all the new amino acids are added to the polypeptide chain. This process starts when the start codon AUG is read, corresponding to the amino acid methionine. The process stops when a stop codon UAG, UAA, or UGA enters the ribosome and allows the polypeptide to be released. After, it might undergo folding to combine with other polypeptides to work as a protein or go inside the cytoplasm.